Welcome back to Divulgando Ciencia. My name is Gustavo Herzhuber. In this video, I'm going to talk to you about a recent research that was made in Switzerland, country where I live. In this study, they explain why some people have a mild COVID while other people have a severe COVID or even die. But first of all, I want to ask you to please like the video, share it with your friends, subscribe to the channel, click on the notification bell to get a notification every time I upload a new video. And please follow me also on Instagram and Facebook. Welcome to this new video. The mechanisms of COVID-19 hyperinflammation are revealed. Research conducted by Professor Michel Gillier, professor at the Faculty of Biology and Medicine of the University of Lausanne and head of the Department of Dermatology and Venereology of the Hospital and University Center from Canton Vaud, CHUV, and Professor Andrea Ablasser of the École Polytechnique Fédérale de Lausanne, EPFL, has just been published in Nature. This is the discovery of the biological mechanism at the origin of severe forms of COVID-19 disease. During a viral infection, the host produces proteins called type 1 interferons to defend itself. Patients infected with COVID who fail to mount this initial defense quickly progress to a severe form of the disease with pulmonary complications. Paradoxically, a delayed interferon response occurs in these patients, leading to destructive hyperinflammation. Published in the prestigious journal Nature, this study shed new light on this paradox and opens new therapeutic perspectives. The role of C-gas sting signaling molecules. The team discovered that the late interferon response leading to hyperinflammation is not triggered by the virus itself, but by the tissue damage it induces through signaling molecules called C-gas sting. The C-gas detects the DNA shed by the dying cells, which penetrates inside the cells and then triggers via the sting molecule the interferon response, leading to the hyperinflammation, a cytokine storm that ultimately generates further destruction of healthy tissue. This is identical to the reaction that occurs during injury and in the context of certain autoimmune diseases. The same research team had already identified this in their work on skin healing and psoriasis. We were intrigued by the fact that bats, which have deactivated the sting gene during evolution to avoid inflammatory reactions during their flight, are resistant to SARS-CoV-2, induce disease and thus constitute a viral reservoir, explains Professor Michel Gillier. This led us to investigate whether sting is activated in COVID-19 events and could be responsible for severe disease in patients. Professor Andrea Ablasser has been studying the sting pathway for years and elucidating the cascade of molecular reactions it triggers. As sting leads to the expression of type 1 interferon and other inflammatory mediators, it made sense to test its involvement in the SARS-CoV-2 induced hyperinflammation process. Analogy between skin and lung lesions. The CHUV dermatology research team first studied skin lesions of patients with severe forms of SARS-CoV-2 and found that they showed significant vascular destruction, with interferon production by surrounding macrophages sensing DNA from dying endothelial cells via the sting pathway. Next, the researchers examined lung samples from patients who had died of SARS-CoV-2 pneumonia and found sting activity in them as well. Using a lung model, the EPFL researchers confirmed that SARS-CoV-2 infection also activates sting signaling and lung endothelial cells as well as in macrophages. The response is then triggered by the release of mitochondrial DNA and leads to cell death and interferon production. Finally, they performed an in vivo study to determine the pharmacological implications of their findings. By administering drugs that block the sting pathway to SARS-CoV-2 infected mice, they discovered a reduction in lung inflammation and a less severe form of the disease. We have identified the C-gas sting pathway as a key factor explaining the aberrant responses to interferon in COVID-19, says Professor Andrea Ablazer. Professor Michel Gillier concludes, our study paves the way for the development of new therapies that specifically block this pathological inflammatory process in severe forms of the disease. As you could see on this video, this study explains why some people get COVID in a light way or in a severe way, and some end up even dying. To explain it to you in simple words, if people produce interferon at the beginning of the infection due to the virus, they would normally evolve into a mild infection, and this is the majority of people. However, if people do not produce interferon, cells, for example, in the lungs are gonna die, and the immune cells are gonna pick up this cell debris. For example, the DNA will activate this C-gas sting cascade, which will induce the production of interferon and then will cause this cytokine storm. If you like the video, please give it a like, subscribe to the channel, share the video with your friends. Please follow me also on Facebook and Instagram. This would help me a lot. Thank you very much and see you next time.